Today we're having a really fantastic celebration because this is actually our 50th webinar. We are really delighted, and I just wanted to take the opportunity to give a big shout out to all of the presenters who have taken part since we set this up in 2019 during COVID to help us all connect and learn from each other. And I'm really delighted as our 50th webinar speaker is Professor Wanda Markotter, who holds a PhD in virology. And she started her academic career at the University of Pretoria in South Africa. She is currently the director of the Center for Viral Zoonoses. And since January 2016, she's also been the DSI-NRF South African Research Chair in Infectious Diseases of Animals. And in 2021, she was appointed as a research chair in People, Health and Places as part of the One Health initiative at the Future African Institute at the University of Pretoria. She also serves as the co-chair for the One Health high-level expert panel. She's a virologist leading a successful research program in understanding the spillover of bat pathogens. Wanda is internationally renowned and very, very well renowned across Africa, and we are absolutely delighted to welcome her today. Thank you very much, Wanda.
Thank you for having me, and it's great to be the one that can do the 50th talk. That's quite significant, and congratulations for that. And I think people really enjoy these webinars. I also listen to some of them. I don't always get time to join live, but because you're recording them, it's always nice to, go back to some of it and hear some of the talks. And it's such a nice diversity of talks that you've had over the years with this. So I'm going to talk about One Health approach to understanding spillover of bat pathogens. I'm going to try and keep it very high level and not go into a lot of detail in our research group. You've heard some talks from some of my emerging researchers. I think Dr. Marika Geldenhuys-Venter gave a talk a while ago on coronaviruses. I'm definitely not going to repeat all of those things, but I'll link it to some of the things that I wanted to discuss today. So because I'm talking about One Health, I thought it's a good idea just to give a bit of an overview of where we are with One Health, and specifically linked to my work as the co-chair of the One Health high-level expert panel. This is a panel that advised the quadripartite, which includes the WHO, WA, who's previously been OIE, and then UNEP, that's part of the environmental organizations, and then also FAO. So it's quite a unique panel to advise these 4 UN organizations together and not give them individual advice. And it's really about where we are with One Health and how we can look at addressing pandemics going forward. And this is just a brief history of One Health. So I think if most of us think about One Health, we think about zoonoses. That's typically where the One Health space have developed, where we look at diseases that spill over from animals to humans. And throughout the years, that's been a very strong focus of One Health. And we sometimes think that is all that One Health is about, that we must look basically at human and veterinary medicine. So if you test animals and you test humans, you do One Health. But it's actually much more than that. And that developed through the years where there was a lot of discussion about how wildlife fits into this One Health picture. There's been different frameworks developed to reduce risk of infectious diseases, where the word ecosystem started appearing in these frameworks on One Health, and we would say that we would look at animal, human, and ecosystem interfaces. But the ecosystem environment interface has never been very strong. It's always been in these documents, but we were not very good at applying it in how we do our research, specifically also looking at prevention of spillover. And then we got to 2021 when this high-level expert panel was established, and this was actually a call by the German and the French government to some of the UN agencies that we need a different way of looking at pandemics going forward, and also bringing in a strong prevention approach. And this is where the One Health approach that I'm going to talk about today is really important, where we're not just looking at pathogens, but we're really looking at a much bigger picture also bringing in the environment and the ecosystems around it, and understanding drivers of disease emergence and not just the presence and absence of pathogens. One thing that OLAP, and that's the abbreviation for the One Health High Level Expert Panel, did when we were established in 2021, is actually developed a new One Health definition that's basically bringing all One Health definitions like planetary, eco health, global health, all of that is in this definition. And this is also very important as a basis when we talk about One Health approaches. This definition has been endorsed by the UN organizations, and it's widely accepted as a new foundation definition for One Health, even in the pandemic treaty discussions. So some of the things we've heard before, where we would say it's an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystems. This is something that we've seen in previous definitions. How do we do this? And this is where this middle part of the diagram is important. It's about the 4 C's: communication, collaboration, capacity building, and coordination. And I think in some of the things that I've seen in the introduction to the start of this webinar today, where you're looking at how to establish BAT groups and all of this, all these things are also important in those contexts. And then it is really about bringing all these sectors and disciplines together, but also different levels of society. And I think sometimes we forget about that part of who needs to be part of the conversations and the collaborations, and who needs to be around the table. So we do recognize that the health of animals, domestic, wild animals, plants, and the wider environment, which include your ecosystems, are closely linked and interdependent. And then it's about the approach mobilize multiple sectors, disciplines, and again, I want to highlight the communities at varying levels of society to work together to foster well-being and tackle threats. And this is a word that we don't always see in One Health definitions. We would talk about the absence or presence of disease, but it's more. Well-being is about more than just the absence or presence of a pathogen. And this is where you can go really broad with things like mental health and other issues that's also part of this. And that we need to tackle the threats to health and the ecosystem in this holistic approach. If we do this, we will also address things like clean water, energy, air, safe and nutritious food. We will take action on climate change, and we will, in the end, contribute to all the sustainability development goals. And I just wanted to give you this context and a few other definitions also that OLAP developed that's starting to inform how we also do spillover prevention and pandemic preparedness responses, the space in which most of my research functions. So I've told you that this definition is really bringing everything together, and we try not to debate definitions anymore and instead try and promote using this holistic new approach to pandemic prevention and preparedness. Another thing that OLAP developed with the definition, and I think this is also important if we develop research projects, is some foundational principles. We sometimes forget about these, where equity is really important between sectors, between disciplines, but also with people involved in the projects. Socio-political parity, where all people are equal and deserve equal rights in opportunities. And this is really where we need to engage communities and marginalized voices. And not just at the end when we think we come with a solution, but it needs to be a co-creation from the beginning if we do work, and also how we do research. Then there's the socio-ecological equilibrium that's important, which also bring in the importance of biodiversity, access to resources, and the value of everything in an ecosystem. The stewardship and responsibility of humans. We need to fix things. We need to make sure that things are going to be better going forward. And then we always talk about a transdisciplinary approach, which means you have to include all relevant disciplines, but not just what we would call scientific disciplines. It also goes as broad as bringing in modern and traditional forms of knowledge, and really also a wide array of perspectives. So this is really very broad, and I will try and highlight some of these things as I go through some practical examples. Then today's talk is very much about prevention of spillovers, and I just wanted to put this in context. What are we talking about? And there's this 2 circles. One is indicated in red, and the other one is in blue. And most of what we do when there's a pandemic or an outbreak, or we focus on this blue part, which is actions to limit spread in the human population. So this means a pathogen is already in humans and we try and use vaccines to stop it, or we do contact tracing and we do certain actions, but it's already in the human population. And where there's a lot of value going forward, because we haven't been very good at preventing spread in human populations, it's really looking at actions to prevent that first case from spilling over from what we would call a reservoir in nature to another animal, and then ultimately to humans or directly to humans. And this is the things that I will talk about today, because this is a true One Health approach where you need to look at risk reduction activities, understand infection dynamics in the natural host. We will talk about bats today. That's been associated with quite a few pathogens, and how we can understand what's going on in that natural host and in the environment. Then what is the contact interfaces? And this is where all the anthropogenic drivers play a role, because the pathogens can be in the bat or in the natural host if it's another animal, and be happy there because they don't make them sick. Yeah, now and then we see disease, but that is not always the case. But it's because there's an interface where it can spill over to other animals and to humans. That's when we have issues, because we become sick and then they spread in these new populations. And then how do we do integrated surveillance, and what do we mean with that? So this is just a summary. This is a paper that we published from OLAP's site that you can also go and read. But there's different things that you can do to prevent spillover from happening. And this also just identify, what it means, and that you need to use a One Health approach. You need to use integrated One Health surveillance. But very important, the second sentence under actions, addressing drivers of disease emergence, also human behaviors, activities that is to do with high risk. But it's also broader things like climate change that influence this contact interface, land use, wildlife trade, food systems, and the list can go on. This is just a few examples. I talked about integrated surveillance, and this is also something that you can go and read that we also did on behalf of OLAP, where we just did a high-level paper of how will One Health surveillance systems, these days people use other words, they use things like collaborative surveillance or integrated surveillance. We're basically talking about the same thing. We still call it One Health surveillance. And what's the steps that you can take? So typically, surveillance in the past has been testing for a pathogen. You test for pathogens in animals or in humans or in the environment, but that is not true One Health surveillance. And if you look at step 2, what is really important is how do we bring in all these drivers that I've just mentioned on the previous slide, and how do we include that in surveillance to be better at predicting risk, but also better at prevention of spillover of pathogens. And that is really what we mean with integrated surveillance. So if you go and test for a vector-borne disease, you also need to bring in climate data, you need to bring in land use data. The same with bats, looking at diversity, looking at land use changes, looking at feeding patterns. All of that is what also need to come into the picture if we talk about truly integrated surveillance, and if we truly want to understand how to look at prevention going forward. So this also some of the One Health high-level expert panel documents that brings in much broader factors than only zoonotic diseases. We wrote this document with the idea that we can update it, and we mentioned several things that you can address through a One Health lens, looking at a problem, all the societal challenges, all the environmental things, all the animal health challenges, also not just looking at diseases, but also animal welfare and other issues that you need to address in a holistic approach. So this is a nice document to look at to try and give you sort of a guideline of how you can develop true One Health approaches around certain problems. So now I'm going to go more into pathogens and discuss that also using some of our examples that we've been involved in. So one of the other things I'm involved in with some of my emerging researchers is the Lancet Commission for Prevention of Spillover. And this is a commission that will do work for the next 2 years with the idea of bringing out a report on how we really think we can prevent spillover in practical ways and give some recommendations. And Raina Plowright is part of this commission with me, and some of you may know some of her papers and this diagram that you can see here on the left-hand side. And this is a very nice framework also for thinking about One Health, because if you think about factors for spillover events, there's a lot of things that actually needs to align for a virus to spill over, let's say from a bat to humans or to another animal and then to humans. And it's not something that happens very often. And all of this needs to align to basically for it to happen. So if we really want to look at prevention of spillover, we need to understand all these factors. We can't just know that there's a virus in bats and then think we're going to do something with that information. We just then know there's a virus in the bats. We don't understand how it's going to transmit. We don't understand any of the infection dynamics. We don't know how we can, you can't make decisions on prevention of spillover if we don't have all the other information. And this diagram has been a framework that we've been looking at some of these things and designing our programs in terms of viral surveillance and also prevention of spillover. You can group it into what is important in part of the ecology and the disease ecology of the source host or the reservoir host. You can look at what is the target host exposure. This is, for instance, where things also comes in like human behavior, the contact interfaces, and the factors that govern infection if there is exposure. And all of this needs to happen. And I'm going to talk to these examples as I go forward, and also explain it on the hand of a Marburg virus example. Now my doesn't want to move. Okay, there we go. This is some of Marika's diagrams that we've also used to explain some of our coronavirus work, and I think she also showed this in some of her previous talks. But there's so many things that you need to bring together to really understand the full picture. And a lot of times we only collect samples from bats and we test it for a pathogen and we say, okay, there's a coronavirus or there's not a coronavirus in it. We may do a little bit of viral characterization, but usually not a lot. And that's where it stops. Some studies will do longitudinal surveillance where you will look over time, viral prevalence has changed or the diversity changed. And some studies will do additional work on full genomes that can help you determine risk in terms of receptor bindings, but very few studies actually do this. Then some studies will look a bit more at the bats. They will do some aging. They will look at birth pulses, when are they pregnant, when are they not pregnant. Some people look at movement patterns, but very few studies actually get to that point. And then what's also very limited in studies is there actually, if we know there's a virus in the bats, do we actually know if it spills over to other humans and animals using serology? Even if we haven't seen disease in a population, there may be some exposures that can tell us what the high-risk interfaces is. And then this top picture, which is basically showing climate and land use and all these drivers, that we seldomly look at. We may bring in a little bit of climate data, but we won't really focus on that. But you can see that if you look at a picture like that, it's expensive. It's a long-term investment. You can't just go into countries and do once-off sampling. You need a big team. You need a big team of expertise also. So it's very difficult in the current funding environment to really put these type of studies together, and very few groups in the world actually are busy collecting this type of data. But I think it becomes more and more important that we look at long-term studies and bigger groups and consortiums to really get good data. So this is the area that we work in South Africa. You may have seen it in some of our previous talks. And this is really an ideal area to look at a One Health approach, because we've got a rural community, lots of agricultural activities. The red is where the houses are, and then the green is where there will be croplands, and then there's livestock moving that's not fenced. So they will be all over this area. And then the one reason we're working here, and you will see this little purple dot, is because there's a large cave here with a maternity roost of the Egyptian fruit bat, Rosetta aegyptiacus, which are known to be a reservoir of Marburg, but also of some of the other potential and known zoonotic viruses in Africa. So this is the area that we've been working in for more than 10 years, starting out with just looking for viruses. And I will show you today, we've increased or expanded to answer some of these broader questions that really requires a One Health approach. There's also other bats in this cave, so it's a mixture of diversity, but we very much focused on fruit bats at the moment. This is some of our work. It actually needs to be updated, where you can see this bats all over Africa, where you see these black dots living in caves, and it's been associated with several viruses, including Marburg virus, which is quite important in our work. So we do longitudinal sampling. We're still going monthly to this area. We can, because we've got a team in country. So it's actually something that's practically possible for us. And we then collect all kinds of samples, urine, fecal, blood, fur. We do our normal viral analysis, but then we also started looking at stress hormones. We try and we pit tag the bats, and we use the two so that we can look at movement tracking. And then we also started putting trackers on that I will talk a little bit about, to look at more daily feeding patterns and also some more long-term. So this is just an indication of what we do. I'm just going to get back to coronaviruses, which is one of our focus area that Marika Geldenhuys is driving. And she gave you, I think, a more detailed talk a while ago. So I'm just going to highlight some things. So we're looking at applying our One Health framework to this maternity colony in this province in South Africa, which is in this area that I've just explained to you, looking at a variety of viral agents, mostly high consequence pathogens, but also including things like rotaviruses and astroviruses, but with a very strong focus on our phylus, paramyxis, influenzas, and coronaviruses. And what you typically get if you do these studies, you get this high diversity that you can see in these trees, and now you need to try and make sense of what this means. Obviously, you can say you've got a high diversity of viruses. In this case, we had different coronaviruses, alpha coronaviruses, as well as novel Rosetta Nubeqa coronaviruses, and then this HKU9 virus that we're currently tracking in quite a lot of detail. It's a HKU9 related virus that has been highlighted in previous studies as a virus of concern because of the high diversity, the widespread throughout the world, as well as a lot of mutations happening in this virus. And if we look at viral characteristics alone, this is things that we would worry about in terms of spillover, because the virus can adapt. And the more a virus can adapt, the bigger the chance can be of that virus going over into another animal and actually recognizing the receptors and spillover. So that is in terms of viral characteristics. It's really just looking at genomic sequences. What we've also done, and this is also where you can bring in things like environmental factors, you can bring in sex, age, reproductive status, body condition, together with your weather and your rain patterns. If you have prevalence data over time, and this is what's plotted on these graphs, we plotted it for all the coronaviruses together, but because we have sequencing data, we could also go into specific viruses and see if we're seeing different peaks throughout the year. At the top, you can see, let me just go back. At the top, you can see our reproductive stages of rosettas. So in South Africa, rosettas only have one birthing pulse, which is a bit different from countries towards Central and Western Africa, where they have two birthing pulses. And our excretion patterns are very much towards certain times of the year where we think there's waning of maternal antibodies of the juveniles coming in. And that is typically what we see for our coronavirus patterns. Now, this is a very simplistic way of interpreting it. We're now looking at other factors like stress hormones. Is it food availability? Does it have to do with environmental stress factors? You can't just say, okay, this is about the juveniles coming in. I think there's a bit more to this picture. And this is typically where you need to do more broader approaches and bring in more disciplines. You can then go further with viral characterization of genomes, full genomes, and also of specific genes. And we've done this for coronaviruses. This is some of our full genome sequences. But what we typically want to look at is, for instance, with coronaviruses, is the spike gene. The spike gene is what recognizes receptors on different cells. And if you have those sequences, just by looking at the sequences, this is not biological systems, so it's just looking at sequences, you can make some inferences of, will it recognize a cattle receptor on a cell? Will it recognize a human receptor on a cell? Then you can make some inferences about risk of potential spillover without doing any lab work or any experimental work. But it's more complicated than that. I think sequence can give us an idea, but in the end, you have to have a biological system to really test if this can happen. But if you don't have good sequencing data and you're only doing short pieces of the genome, you can say nothing about this. You can just say it's related to another virus. So it's really important that you get to the point where you really characterize what you get in terms of the viruses as the first step that you do. Then another thing, this is some of the work that Marika presented in Cape Town. I see I've left it at the bottom. If you have tools to look at antibodies, you can do quite a few nice things in this broader picture of not just looking at bats, but also looking at potential spillover to livestock and potential spillover to humans. Because you will probably not see disease, you want to test for antibodies, for instance, in livestock to see if they've been exposed to a coronavirus. And you can then make some inferences about contact interfaces. The same with humans. And in the past, and still, we have very limited tools for serological surveillance in bats and then also in spillover hosts. Because the viruses are not the same, then they're related to what we see in humans. We don't have commercial assays. We need to develop all of this to really also limit your cross reactions, to have a specific antigen that you use in the test. And this is what we started to do now with our coronaviruses. We made specific antigens for HKU9, the virus that I showed you that we've detected. And then we can track serology over time and see how that actually correlates to specific times that we can see excretion of the virus. And this can tell us things about duration of immunity in populations, but also the immunity levels. How many times do the animals also need to get exposed to actually develop protection status? So this is really preliminary things, but you need to have the tools to be able to do this. And that also limits a lot of the type of studies that we can do. So we come back to what? We detect this diversity of genomes. We can identify them. We can say they're related to some highly zoonotic viruses, but it's not the same virus, for instance, as SARS coronavirus 2 or a virus that we know in humans. So what does it really mean? So there's different ways that we can look at it. We can start doing really fancy mathematics and risk assessment tools. Spillover risk ranking tool is one of them. And I've mentioned to you that HKU9 has been, according to this information, ranked as number 40. Now, this is information that we just take from studies and we throw it into these tools and then we model them. We make a risk assessment. So it's not based on real data where we actually know the virus has made somebody sick or this is a spillover route. So it's all predictive type of work. We can also think about, as I've mentioned, the potential of the virus to infect other hosts by using animal models, cell culture. So you can take the viruses, put it into other systems, see if they make them sick. You have to have the virus isolated for that, which is extremely tricky with bat work. Firstly, because you need usually biosafety level 4 labs, but also because these viruses don't even want to grow. We usually only have nucleic acid data. You can look at what I've showed you now with the sequencing data at specific characteristics in the virus to make some predictions on if it will recognize specific cell types. And then the serology also, as I've showed to you, if we have those tools, we can look at maybe there's been exposure, and that exposure may at some point lead to a disease and infection transmitting between animals or between humans if we know that contact interface is there. So we have done some of this for years, where we understand viral genetic diversity. We even know quite well about the mutations that's occurring and how these viruses has changed over time. We know quite a bit about how it's maintained in the host, specifically our Rosetta's work. We understand quite a lot on how it's maintained, and the infection over time will be maintained in these hosts, and what may be the factors, age and stress and all of that that's driving it. We also know that there's co-circulating viruses, and we sort of know the timing of opportunities for spillover. And this goes back to the peaks that I've showed you, where sometimes in the year there's almost no virus. So we collect fecal material for coronaviruses. So we test fecal. Corona as a gastrointestinal virus in bats, but as soon as it goes into humans, it's more a respiratory virus. But in bats, it's gastrointestinal. So you can collect fecal and you can look at the prevalence over time. And we see certain times of the year, the virus is almost gone. Other times of the year, you have these high peaks. So we now have an idea of what is a high risk period, because if you look at opportunities for spillover, it will be when the virus is there and when the virus is potentially in a high prevalence. So we can now look over those times at what can be the interfaces for contact. So we can look at serological testing of other animals over the year, and specifically domestic and wild animals, but also humans, but then also try to look more at the anthropogenic activities in the area to determine what the drivers may be that will influence potential spillover, but also stress in bat populations that may lead to higher prevalence of viruses. So this is the ranking tool that I've talked about that was developed by Franche et al. And that's been published. And a lot of people are looking at all these factors that's mentioned in the spillover ranking tool. And you will see, I'm trying to move the chat so that I can see my slide. There we go. You can see that there's a lot of factors on the virus, but then there's all these interfaces that they mentioned in the spillover ranking tool that you can look at: livestock density, deforestation, urbanization. A lot of times we have no data on this. So we actually just need to say, sorry, we don't know. We can't make a prediction on risk on some of these factors because we just don't have data. And if we do have data, it's usually global data that we're modeling to other parts of the world instead of having data for a specific area. And that's not necessarily the best way to do it. Some of the things that we brought in specifically to look at interfaces is trying to look at movement patterns. And this was some of the work of some of my previous postdocs, Flo de Vries, where we looked at contact with human, livestock, and wildlife interfaces. We tried to look at diversity of wildlife. We looked at fruit abundance and fruit trees in the area over season. Also some diversity work with sharing with other insectivorous bats. And we're using very basic methods, just looking at camera trapping, what is getting in contact with the bats, humans, other animals, so that we can design future studies to look at potential spillover species. We've also done some movement work, first just with radio tracking and then later on with some GPS tracking. It's a bit complicated with Rosettas. They live in caves, so you can't use solar panels. So immediately you have a limitation on the size of the devices, which also links to the amount of data you can get over time. So we can typically, with a GPS device, only get 3 days worth of data. So we can't look at long-term movement like you can with Eidolon and some of the bigger species, just because of the limitation in devices at the moment. With our radio tracking, we looked at annual feeding over a whole year. And if you look at the red areas, this is where the housing is. And in the summer, they feed in the natural areas. From observations, it's usually the wild fig trees. But then in the winter, if you look at these yellow spots, they moved into the human dwelling areas and actually feeding on fruit trees that's planted in these human dwellings, like mangoes and other type of things. So immediately in the winter months, there's more interactions with the human areas in this specific area. And this is also the time that we're seeing higher excretion of viruses. So in terms of that, this is giving us data to show these high-risk periods and potential interfaces by feeding patterns of bats that change over seasons. This is some of our satellite, our GPS tracking data, where we could track individual bats to specific areas. Some of them still go and feed wild fig trees, but some of them went into human dwellings feeding on a specific tree where there's interaction with domestic animals, dogs, but also with people, and a lot of fruit also dropping to the ground that can actually be picked up by people, and there may be saliva contamination from the bats who was feeding on it and dropping the fruit to the ground. This is a paper from CDC that brought this interface of potential fruit getting contaminated with saliva. It sort of made that a reality that we had to think about. So this is experimental work where they put Marburg virus on fruit in the lab, and they would see how long the virus stays viable. And it was up to 6 hours. They didn't go longer than this. So it can actually probably be longer than this. But this tells us that if a bat feeds at 4 o'clock in the morning or 2 o'clock in the morning and it drops fruit to the ground, a pig can pick it up or a human can pick it up, and there may still be contaminated saliva that has got live virus in it. So we have to look at this interface of fruit and contact with other species and humans. We know that Marburg virus are also present in saliva. It's present in saliva, urine, and fecal, actually. So that is a potential way of the virus transmitting. Some of the other work that we've done in South Africa to also look at these interfaces, and this is very preliminary work, was looking at how land use are changing around our bat caves. So we mapped some of our important maternity roosts in the country, and we looked over a certain amount of years of how land use has been changing. And it was quite shocking how urban areas are moving into these buffer zones. Basically, well, we don't have a specific buffer zone around a cave, but that area is really becoming much smaller, which means bats will have to go and feed somewhere else, will have to fly further, and there will be other contact interfaces. And it's also just not good for bat conservation. So we made a few recommendations on how we should address these issues. So really understanding human activities and the contact that drives spillover is really important. And this is just a few pictures, some of them taken still by my colleagues that did some Ebola work, Bob Swanepoel and Janusz Paweska. These interfaces are very different. It's not the same everywhere in the world. This is a picture, the one in the top right corner is from South Africa. This is a religious interface where people still go into caves for religious purposes in South Africa. They spend quite a bit of time there, sometimes even sleep there overnight. There's some eating and drinking in these caves. So immediately you have this exposure interface. Mine's always an issue. Sometimes catching bats and eating them, hunting them for wild meat, putting them into markets, deforestation, intensified farming around bat interfaces has also become quite a huge issue, where we have a mixture of bats and more intensified farming of domestic and sometimes even wild animals that may play a role. And we need to understand these contact interfaces in context. And that goes together, I think, with something that you are very good at is really education. You can do all these things and say, okay, at the end, I'm going to decide on what I'm going to tell people. But it's a continuous process as you do work to develop educational material, to have interactions with people based on the knowledge that you're getting throughout your research, to try and prevent people from being exposed to viruses, but also addressing the broader issues of just conservation of the area and conservation of the species as we have more information. So I'm going to end off with Marburg, and this is also work that we've done, we're busy with through the Lancet Commission, and I think this is very topical at the moment, and how we're starting to think about research on Marburg virus across Africa, and specifically the cave mine interface. So I'm not going to talk here about the fruit interface, but specifically direct contact with bats and then getting infected with Marburg virus, and this leads to human outbreaks. I haven't updated this map we've now got the outbreak in Rwanda, which is a new country being implicated. Also, potentially with an index case where there was a direct contact with a bat, and then it started making healthcare workers sick. So going back to that slide that I've showed you from the diagram from Rainer Plowright on what we need for a spillover to happen. So for instance, if we look at Rosetta Egyptiacus and the source host distribution, this is a map of museum records. We know the bat is distributed through various parts of Africa. It actually goes into the Arabian Peninsula also. Patchy because they live in caves, so that's why you will have, it's not continuously distributed. We know things like they move between seasons. They definitely don't stay in one roost, so you're definitely dealing with meta-populations. We also know that they will move according to food availability and also their reproductive season. So they have their maternity roosts and not all the bats stay there throughout the year. So we try to look at evidence and then identify research priorities where we still don't have enough data, like on the host distribution, and the ecology is very outdated, as well as how these populations are connected, mostly also because we don't have very good tools on looking at movement of Rosetta Egyptiacus. There's also, quite interestingly, and this is very important for us looking from a prevention of spillover perspective, where natural roosts have been damaged or closed, like cave or mines, they have moved to anthropogenic structures. So there's been reports of them moving into roofs of houses in Northern Africa, which is quite concerning, seeing this is a Marburg virus reservoir. If we have these interfaces changing, that can lead to more contact and more outbreaks and contact between humans. So one of the things we're trying to do, and I'm not going to go through all the detail on all my slides, but what is driving this? And this is where the questions become very complicated and really require a One Health approach, because we can say all these things, but we can tell people, yes, okay, they can't be in a roof of a house, but what is driving it? You know, what is driving the contact between bats and people directly? Things like ecotourism, mining, maybe guano mining. There's so many things that's driving that we need to address if we really want to have a solution for addressing these interfaces and really getting to a prevention point. But we also know in terms of density, we know a little bit about bats and how they move, but we still don't know, like I said, everything about the meta-population. What we do know as a proximal driver, if we cull back and we exclude them from the habitat, and that has happened, it will increase the viral prevalence. There's been a very nice study by CDC that as soon as you put bats under pressure, it's actually going to come back in much higher prevalence, the virus. So although people feel like they want to cull bats and exclude them from the natural habitat sometimes, and it does happen, it leads to this higher amount of specifically Marburg virus presence in these populations. So we know quite a bit about how the prevalence of Marburg look in populations. It's not very high, 2.5 to 5% in normal healthy Rosetta's populations. But then what we also know is if there's stress on the bats, it goes up to 13% versus 5%. So stress on bats definitely have an effect on the amount of prevalence of virus. And this goes back to how we deal with bats in terms of conservation, in terms of bat health, in terms of giving them their natural, or keep their natural environment so that they're not under stress, because then we will actually see more of the viruses. So that's also some of the things that we need to look at. We don't know a lot about if the intensity of infection differs over time. There's been a few experimental studies, but we don't know this. And why is this important? Because there probably needs to be enough virus for you to get sick. So if the titer or the viral load is very low in a reservoir, and we see that with some viruses, that's probably why we can't even grow them in the lab, then there's probably no risk because it's not enough to make you sick. So it's quite important that we also have an idea of if there's time periods where the viral intensity is higher. We need to know how the pathogen is released from the host. And with Marburg virus, we know it comes from urine, fecal, and oral swabs. We've been looking at this in experimental as well as natural colonies. We could identify it. We also know that shedding occurs for quite a variation of a period, 3 to 19 days. We know that the virus survives outside the host, so that interface is there. I've told you about the fruit infections that went up to 6 hours. And then there's also the possibility of aerosol transmission and survival that we have not been investigating very well, where we have very humid case. So all of this needs to drive, this will drive the prevalence, which will affect if there's a risk of spillover. So some of these things may be linked to temperature and humidity, survival in the environment and all of that. We don't really understand that or have any evidence of that. So climate may play a role in the risk of spillover. And then we get to things which is also very true to a One Health approach. What is the target host exposure to the pathogen? We know it with Marburg. We know it's been linked to miners and tourists entering caves and mines where this bat has been present. So the driver there is really, what make people go into these natural habitats? Mining, ecotourism, guano mining, and what is the reason that we will have these interfaces? And then it's definitely about the contact with the excreta in these mines. I've told you it's in urine, fecal, and all of that. And then also, do we know the dose or the route of exposure to humans? We know it's present in fecal, urine, and saliva in bats, but what was that specific route of exposure to humans is not always very clear. So this is a very complicated slide that I want to end off with, and we're still also wrapping our minds around this. We've got all these factors that's important for prevention of spillover that we need to understand. And we can't really understand them in isolation, and we can't really skip any of this understanding, because then we don't have the full picture if we want to get to a point where we want to make recommendations on prevention. And then you've got all these other things that you can see in orange and green and blue that's driving this. That's probably the important things that we need to understand to actually get to mitigation actions that will work. Things like land use changes, the environment, indigenous and local traditions. You can't just say it's a factor that's influencing it and stop it. You really need to understand all these things and also understand if it's got a real effect on the viral spillover, and if it will have influence if you bring in some mitigation actions. We basically have almost no data on these factors that's driving all of these interfaces of interaction. We have, with Marburg specifically, quite good viral data, although there's also some real gaps in ecology that we don't understand. But there's also still a lot of question marks on some of the outbreaks. How did it happen? Where there's no identification of a direct human bat exposure. How did humans in the end get infected? We have quite good data if we talk about mines and caves, but there's also been human cases where we have no idea where the contact interface was in terms of Marburg infections. So it's really a much bigger picture to understand, not just focused on virology, and really thinking about One Health if you're thinking about all of these factors, which is about animals, humans, and then the whole ecosystem. That is important to understand the full picture. So it really comes back to what I started with what we believe One Health means, what it means to implement it, who needs to be around the table, and trying to develop programs to understand this bigger picture over time. It's not a quick process, but at least trying to ask the difficult questions that's broader than just doing the virology. So I'll end there. Thanks. Any questions?
Excellent. Thank you very, very much, Wanda, for a really informative talk. And it's always the case, kind of the more answers you have, sometimes the more questions that then brings about. So we've got some questions. So a nice presentation. Thank you, Prof. With the evidence of bats carrying viruses, how do you answer questions that somebody asks about bats potentially causing COVID-19 pandemic, especially if they have little scientific background, and you're obviously trying to get them to appreciate bats? And I think that's a really good question that a lot of people probably struggle with, is get that balance between, kind of honesty and scientific evidence, but also putting it in context of, kind of bat conservation as well. So trying to get that balance.
Yeah, it's always a difficult question, but I think my answer always is, so bats don't get sick from these viruses. They've learned to live with them. It's probably been evolving with them for hundreds of years. So if the bats and the viruses stay there, they will be happy, everybody will be happy. And it's really about that interface. And so we need to think, if we interacting responsibly with wildlife, and that includes bats, we shouldn't just go and catch bats without the necessary protection. If we're in that environment, it's not a good idea to do that, because you're exposing yourself. If we want to do things like guano mining and it's approved, then we need to have certain protection. I mean, if you work at a mine, you wear certain protection. If you do things like that, there's certain risks that you need to think about. And I think it's really highlighting that interface, specifically thinking about COVID. So there's related viruses in bats that are related to SARS coronavirus 2, but the virus didn't spill directly over from a bat to humans. So it's again about that interface. And what we think that interface is these animal markets where animals are kept together, mixed, in stressful situations, urine and fecal all over. So that is what we need to address, and what we need to be worried about, because that's when the viruses are going to amplify and they're going to spill over to people. So it's really, I always steer the conversation to that interface that's not right, that we're causing, instead of the virus that was very happy in the bats. It's really about what we do.
Yeah, that's great advice. So we've got another question saying, also, thank you very much for your presentation. With your experience, do you think viral prevalence is higher in cave bats than those living with fruit bats? I'm assuming that this question is meaning not, fruit bats not living in caves, but maybe you could compare fruit bats in caves and not in caves, and cave-dwelling insectivorous bats.
Yes, I think my gut feeling from data that I've seen, I would say yes, and there will be a few reasons for that. The bats in caves are usually much larger colonies and they're on top of each other. So it may just be, the ecology of the bats and easier transmission. It's also been a bit biased. It's easier to catch bats in caves, so we've done more work. And you can catch higher amounts very quickly with a half trap compared to doing mist netting over a large area, for instance, of other species. So in South Africa, we've never done a true comparison just because of that reason. But I would think it's also the ecology of the specific species.
Yeah, and I think that's the same, actually. If you look at museum records, it's definitely biased towards caves, because it's just easier for trapping, for sure. So so we have a question here saying, could you kindly shed more light on how the Rwandan Marburg outbreak started?
So there's no official data out, so we don't really know officially, but there's some Twitter comments and things that it was, the index case was a direct roost exposure, but then it amplified in the healthcare setting. So I think all the subsequent deaths were healthcare workers, but it seems to again be probably entry into a mine or into a cave, and started from there with somebody getting sick, going into hospital, and then because of poor infection control, that spiraled out of control.
Excellent. Okay, thank you very much for that. And so another question with a fellow virologist saying, I'm currently working on a project investigating arboviruses in cave bats in Burkina Faso, and I'd like to know whether using blood on FTA cards also gives better results than blood taken from tubes.
So it depends what you want to do. So FTA cards can inactivate viruses, and that can also damage antibodies sometimes. So we've been playing around with that. We don't really have data exactly with our systems what will work best. But you're always sitting with a situation, if you just collect blood without any inactivation method, you're sitting with a potential infectious risk. So it becomes really difficult for people to collect blood in tubes without inactivating them anyway with heat or other methods. And your FDA cards, it depends on what you use, can inactivate viruses. So immediately you're not sitting with that. So I think we need to be better at looking what we can use these inactivated type of sampling methods for. And then with FDA cards, you need to be careful that you have a way of standardizing the amount. So if you want to look at titers in serology, for instance, if you're doing serology, if you're just doing nucleic acids, that's not an issue. Because your dots need to be consistent and how you dilute that. So there's all kinds of practical things that I can discuss with you. We've lately been looking at that quite a lot.
Excellent, thank you very much. And then we've got another question. Based on your experience with Rosetta-borne viruses, is there seasonal determinants strongly associated with the timing of the virus spillover? So I must admit that was one of the questions I was interested in asking you as well, because I'm guessing obviously when you were mentioning kind of winter being the problem, and I guess there's maybe a higher stress, and you had some hibernacular or hibernation sites. And actually, I guess if there's there's sort of food availability and higher stress, because you kind of almost think in some ways that actually during the breeding season would be quite a stressful time as well. So yeah, if you have any insights into kind of the sort of the timing of the virus spillover.
Yes, so the work done on Marburg in Uganda by CDC actually correlated that very well. We can't do it in South Africa because we've never had a human outbreak, although we've got Marburg virus in our bats. So we could never say, the same time that we saw people getting sick, we luckily never had human outbreaks. But in the Uganda situation where they have Marburg in bats and Marburg in humans, they could time it with these high prevalence periods, which they linked those years also, as we did, just to reproductive seasons of bats. When you have your juveniles losing maternity immunity, and you probably have a higher infection peak. And that's how simple we left it. But I do think we're now looking more at stress and food availability and all of that, but we haven't timed that very well. But it may be the same periods. And it was linked also to people going into caves during those periods when there was higher amounts of virus being shed, probably in urine and fecal, and getting into contact with those viruses.
Yeah, and there's more questions coming in, but I'll just finish off with this last one, because I guess it's kind of the I suppose, as you had spoken about, there's obviously kind of habitat fragmentation and loss, kind of really are, kind of linked more to zoonotic diseases as well. So I guess, how do you see kind of our role? And I get, like you had mentioned, we really all should be working towards prevention. So I guess sort of our role in sort of, I suppose, habitat protection, but also enhancement. And as you have been doing that advisory role, because it's, the more we are damaging kind of the natural environment, species are having to come into more urban settings for their survival because of a situation we've caused. So I suppose if you've got, I suppose how do you find that the One Health fits in with that sort of, kind of ecosystem sort of protection and health and restoration?
So I think it's integral in the ecosystem part of One Health that we need to look at that. I think what's really needed is data, because people don't just want to react on what we tell them, even if it's our gut feeling and we know it. We really need to start monitoring these things over time to show change. We need to monitor severe events, where we know things have been damaged. And then we also, what will be great is during these times have some risk of pathogen prevalence also to convince governments or conservation agencies that this is really important, not just because of the tree that you cut down, it's because of all these other stress that you're causing, and in the end Marburg virus is higher. And I think also you can play a very important role in the type of interfaces that you see in terms of interaction with humans. It's very different, we've realized, in different contexts and the reasons why people are doing it. And you may have a more trusting relationship than a virologist going into those areas and trying to mitigate those interfaces that's high risk. and working with communities that you have relationships with to try and change that behavior together with them towards a better behavior.
Yeah, no, that sounds great. And that's actually also, it's a nice positive. That's something we can all be working towards that. And I think you're right, actually. And it's really nice to see such interdisciplinary research and how much, and I'm also very jealous of all your your tracking data. I must admit, that's, even if you are, it's limited, but it's still really, really fantastic data. So I just wanted to take the opportunity to thank you very, very much for coming along and giving us such a great informative talk. There's more questions that we can cover today. So thank you very, very much, everybody. And if anybody didn't catch Wanda's email at the start, which was on the slide, do get in touch with us, or obviously you can watch the video again and catch up on all the information. And obviously there's lots of great papers as well that you can take from this webinar and look those up. So thank you very much, everybody, for your time, and a huge thank you, Wanda, again, for coming along.
Thanks. And if there's any more questions, just drop me an email. I don't think we got to all of them. You're welcome to contact me.